Alright guys, today we're going to be talking about DNA structure. Alright, so DNA structure, let's get right into it. So it all begins with a cell. And in this case, we're using a human cell. And this particular human cell belongs to this guy over here named Jim, right? And Jim has trillions of these cells all over his body. They make up all of his organs, all of everything, pretty much. And this right here, this blue part right here, is the cell nucleus, and it's basically the brains of the operation, and it controls the entire cell. Now inside the cell nucleus we have many chromosomes, and chromosomes are made up of multiple different components. First is the centromere, right, which is the center of the chromosome. So first is the centromere, the center of the chromosome. Then we have the telomeres, which is the ends of the chromosome. It could also be the top ends. And then we have the chromatids, which are the half and half portions of the chromosome. And the chromosome is basically composed of many strands of DNA, right? And those DNA are held together by proteins called histones. And then when we have multiple of histones, when we have 4 pairs of histones, right, those pairs create something called nucleosomes, right? And we're gonna show you that nucleosomes are nucleosomes, what are nucleosomes, right? So nucleosomes are the fundamental repeating unit of chromatin. Chromatin is essentially a lot of DNA that's inside of a chromosome. A lot of DNA inside of a chromosome. Now, these 8 histones that a nucleosome consists of contains 2 copies of each different type of histone. And they are called H2A, H2B, H3, and H4. And they basically wrap around, or the DNA wraps around them and creates this coil. But you're probably wondering, Tyler, what is DNA? Right? What is DNA? Right? Well, we're going to show you right now. Here's a model of DNA right here. First thing we got to understand is DNA stands for deoxyribonucleic acid, right? And we have our DNA molecule right here. All right, so it's a molecule that contains the genetic instruction for development, functioning, and reproduction for all living organisms. But you're probably asking, well, what is that in simple terms? Well, basically, it carries the blueprint for a living organism, for a living creature. I would rewind and try to memorize this term. But anyways, DNA contains 2 components. We have the nucleotides and the phosphate backbone, right? And together they form a double helix shape. They go in opposite directions, and we call that anti-parallel. And they're held together by hydrogen bonds, all right? Those are the key ideas that I want you and you should remember about DNA, right? Now, DNA can later on be copied, right, and become something called RNA. Right, RNA right here. This is RNA. And RNA consists of a single strand. There's only 2 differences about RNA, and I want you guys to remember this. It's a single strand, and it changes one of the nucleotides from T, which is thymine, to uracil. So remember that. Now let's get into the structure real quick of what is like the whole DNA. Let's like zoom in and see what is the 3 parts of DNA. DNA has 3 parts, right? And those are the sugar, phosphate, and base. So the sugar is in the shape of a pentose, right? A pentagon, pretty much, right? And that pentagon connects to a phosphate group, right? And then that pentagon also connects to a nitrogenous base. And that nitrogenous base can be something which we call a purine or a pyrimidine, right? And that's basically the A, T, C, G, or U that we will get. So remember, it's a phosphate backbone, pentose sugar, and nitrogenous base. Those are the 3 parts of DNA. Now, what about those nitrogenous bases? So these are the nitrogenous bases types I was talking about. So there's 2 types. We have purines and pyrimidines, right? And I know it sounds, it's like a big word. Trust me, I know, I've learned this. So purines can be either adenine, A, or guanine, G. And pyrimidines can be cytosine, uracil, and thymine, right? So CUT. But uracil is only for RNA. That's a key rule right there. Only for RNA. We don't really use uracil for DNA. And uracil replaces the thymine. It goes in the place for thymine, right? And thymine is only for DNA, likewise, right? And the connections we follow is this formula called, or this rule called Charkov's rule, right? Charkov's rule, right? And basically that states that adenine will always bind to thymine and cytosine will always bind to guanine, right? And this is only for the A and T though, is only in DNA, right? But if it's in RNA, like I just mentioned, A will always bind to U. But that's basically the idea of Charkov's rule. A binds to T in DNA, right? C always binds to G, both in DNA and RNA. And then A binds to U in RNA. Okay, so let's go over and review what we just discussed here about the DNA structure. So DNA is in the shape of a double helix, right? And that's because it's anti-parallel, right? And contains complementary nucleotides on each side of the phosphate backbone. And we know that DNA contains 3 components, which is the phosphate component, right? The phosphate base. And then we have the pentyl sugar, right? And the nitrogenous base. And the nitrogenous bases can be either purines or pyrimidines, right? And T for thymine, only for DNA, and uracil is only for RNA. And those 2 nitrogenous bases are held together by hydrogen bonds. And this is the zoomed in version, right, of the same model I showed you guys earlier. All right, the last thing we're going to go ahead and talk about here is something called a DNA hairpin, right? You're probably like, what the heck is that? I know, I asked the same question. But a DNA hairpin is a structure that folds on itself, a single strand of DNA that folds back on itself and forms 2 components called a stem and a loop. And this can be used for basically regulating gene expression and other biological processes, including it will be used in transcription, which I'll explain to you guys later on in another video. But essentially, just think about that the nucleotides have an affinity to bind, right? So the nitrogenous bases, right, have an affinity to bind, but they have an affinity to bind not because of necessarily always their own affinity, but because something else is influencing them. So just remember, it's a strand of DNA that folds in over itself, forming a structure that looks like a hairpin or a stem and a loop, right? So it forms a stem and a loop structure, or otherwise known as a hairpin. So that's all you need to remember. So today we talked about chromosomes, basic chromosome structure. We can go too deep into it. DNA structure. What is like DNA? What is the purpose of DNA? And what is a hairpin? Briefly, what it just introduced you to hairpin, right? We briefly just introduced hairpin. In the next video, we'll talk about DNA replication and the beginning of transcription. Thank you guys for watching. Hope you guys learned something. If you have any questions, go ahead and put them in the comment down below, and I will be happy to answer. Thank you.